Good afternoon. My name is Rukaya and I will be doing a case study on the Human Genome Project. The Human Genome Project was an international scientific research project with the goal of determining the base pairs that make up the human DNA, identifying, mapping, and sequencing all the genes in the human genome. The Human Genome Project started in October 1990 and was completed in the year 2003 in April. It took researchers 13 years to map out around 3.1 billion chemical base pairs, namely adenine, thymine, guanine, and cytosine. The human genome has around 20 to 25,000 genes. The whole project cost around $3 billion. The HGP was initiated in 1990 under the leadership of American geneticist Francis Collins, with the support of the US Department of Energy and the National Institute of Health, the NIH. It was a collaborative research with the countries like Japan, Canada, China, Nigeria, the US, and the UK. The process of identifying the boundaries between the genes and the features of raw DNA sequences is called genome annotation and is in the domain of bioinformatics. The strategy is called the hierarchical shotgun sequencing, which involves many independent steps. Firstly, large fragments of human genome were cloned using the BACs, which is the bacterial artificial chromosome methodology, in which we use bacteria as the host. Human DNA was fragmented into pieces of around 150,000 to 200,000 base pairs each. These DNA fragments were then inserted into the BACs and were cloned in the bacterial cell, which replicate the human DNA fragments to obtain enough material to be sequenced. Once the DNA was replicated, the DNA fragments were randomly broken into smaller pieces to be sequenced through the automated Sanger sequencing. The genome was published by the HGP does not represent the sequence of every individual genome. It is a combined mosaic of small pieces of anonymous donors of African, European, and East Asian ancestry. The HGP does not provide the scientist with powerful new approaches to understand and development of diseases and to create new strategies for the prevention and treatment. Nearly all human medical conditions except physical injuries are related to the changes or mutations in the structure and function of the DNA. So it is quite information to know the entire sequence of the Human Genome Project. Thank you.